Over the next 4 or 5 weeks, we're going to be working on DNA and RNA biochemistry. And it's important that we go over some of the things that you learned in other classes in 461 and your biology classes. I'd like to start with the term central dogma because that governs the biological processes that are used by DNA, RNA, and protein to allow the expression of genetic information into the kind of machinery that we need to support cellular life. So DNA, of course, is what encodes genes, and if that information is copied from one generation to the other, we have to copy that DNA very faithfully. We call that process replication. The copying of that same genetic information into RNA is called transcription. RNA serves as a short-term, temporary copy of that information, and the purpose of it is to ultimately guide the production of the protein that is encoded by the gene. And lastly, the process of converting the genetic information in RNA to protein is called translation. It is named that because we are translating from one language, that is the nucleic acid language in RNA, to a different language, the amino acid language of protein. Okay, at this point I'd like to do a little review on the basics of DNA structure, with a particular emphasis on the DNA backbone. You may remember the components of DNA are first the deoxyribose sugars, the nitrogenous bases such as A, G, and C, and T, and finally we have the phosphate groups that connect the sugars together. We're going to be learning a lot about how these components actually come together in the cell, but for now it's probably enough for us to just say that we need to remove waters and then connect what's left over. That's going to look something like this. Now here's a couple of important things. This phosphate connects the 3' hydroxyl of one ribose to the 5' hydroxyl of the next ribose. There is a COP double bond O phosphoester and another COP double bond O phosphoester, making a 3' 5' phosphodiester linkage, or PDE. We should also note that for every phosphate there is exactly one negative charge. That'll play an important role later. Probably the most important thing we can learn about DNA backbone structure is the issue of directionality. And that rises from the fact that we have in every DNA two termini with free unpaired alcohols. On one end we have the 5' alcohol free, and on the other end we have the 3' alcohol. And the key is that by convention, DNA always goes from 5' to 3'. It's so critical that you understand this. I would also like to point out that sometimes we can't see the termini of DNA, but we still have to figure out directionality. There's a simple way to do that, and that is to recognize that within each ribose of the DNA structure, there is a 5, 4, and 3' carbon. You can see them here. The directionality from 5 to 3 within a ribose matches the overall directionality of the entire molecule. Okay, next we need to move on and talk about the fact that DNA is a double helix, and directionality is going to play a big role here again. Let's start out by pointing out that DNA, of course, is going to be 5' to 3' on one strand, and in the opposite direction, or anti-direction, 5' to 3' on the other strand. Next, we can note that because of the phosphates in the backbone, both strands have a high degree of negative charge, 1 for each base and 2 for each base pair. This is going to play a big role in our DNA structural issues later on in the class. Next, I'd like to point out that the bases are going to emanate out from the backbone in between the phosphates and reach across to the other strand. We can see them here, coming from both strands, where they contact each other in between. I suspect you remember that in DNA, the bases are held together by hydrogen bonds, and we'll talk about that a little bit more on the bottom of the page. They're certainly extremely important. However, we need to get a little more into the chemistry here and talk about other stabilizing factors. Because the bases themselves are aromatic, we're going to have pi clouds above and below the rings. And those pi clouds are going to interact with each other and help stabilize the bases from one strand to the other. So these pi stabilizing forces are going to be called stacking forces. And they play just as big a role as the hydrogen bonds do, so we have to make sure to note them. So everything we see on the left side of the page is true on the right side when we twist up the two strands in the double helix itself. We have anti-parallel 5' to 3' directionality. We have the negative charges. We have the bases reaching across from strand to strand. There are two more important ideas that we need to get to when we look at the double helix. And the first one is that it is right-handed. What that means is it has the same chirality or screw sense as a normal wood screw or metal screw that you would screw into the wall. The other thing we want to note is that the two strands do not come together in a symmetric fashion. Because of that, we end up with grooves between the two strands that are not the same size. We can see a gap between two strands that's fairly small and shallow, and then there's another gap between alternate strands that is wide and deep. As you can imagine, we call these things by descriptors that fit. The large one is referred to as the major groove, and naturally the small one is therefore the minor groove. It's really important that you can recognize the difference between these, so this is something we'll definitely practice on the homework. We're going to talk a great deal more about hydrogen bonding between bases at the bottom of this page and on the next page in class. So at this point, I'd like to add two pages to page 5 and just finish up with Chargaff's rules. Erwin Chargaff in the 1930s discovered some interesting observations about double-stranded DNA. This was long before Watson and Crick discovered the structure of DNA, so the implications were not obvious at the time. But they were so fundamental that it's worth talking about now. Chargaff discovered in double-stranded DNA that the content of A, that is the percentage of A, in any double-stranded DNA, always matched the percentage of T. He also noted that the percentage of G equaled the percentage of C. Now the implications of what we see here may be obvious to us now. A is equal to T because they base pair together, and likewise for G and C. But at the time, nobody knew what these meant. Chargaff's third rule is very interesting and also not quite as obvious. It turns out he noted that the percentage of A and the percentage of G is always equal to 50%. Likewise, the percentage of C and the percentage of T also added up to 50%. This rule stems from the fact that A and G are both purines, and since every base pair must have one pyrimidine and purine, The total number of purines you have must always be exactly half of the total bases. Pyrimidines, of course, are C and T, and they must represent 50% as well. What's interesting about Chargaff's rules is that if you know the percentage of any one of the bases in double-stranded DNA, you can determine the percentage of all three of the others. This is a fact that we'll exploit on another homework exercise just to make sure that you understand how these rules can be applied.